Thank you, Nina. Hi, everyone. Yeah, thank you for organizing this. It's great to be talking to you today here. Thank you for the opportunity. I would just briefly present myself. So I am Suzana. I am from Brazil, from Rio de Janeiro. And over there, I studied oceanography, and also I did a master's on plankton ecology. And that's when I got very interested about the diverse trophic strategies that plankton can have, and also about all the complex interactions within the microbial food web. So I got to think about how we could use models to understand these systems that are so complex. And that's how I crossed the Atlantic and I went to the UK to do my PhD. So over there, I collaborated with researchers in Swansea University and also the Plymouth Marine Laboratory. And we were particularly interested on mixotrophs. We wanted to investigate their biogeography in the global oceans and also how they could impact ecosystem functioning. So if you are like I was and you don't really know where Swansea is in the west coast of the UK, and nature is pretty impressive there. So this was my expectation when I first flew to Swansea, but that's what I got. A lot of rain, wind, and cold weather. So after 4 years, I said, that's enough of this climate, and I really need a new research challenge. And that's how I met Naomi, and then I came to LA. So now I am a postdoc at the Levine Lab. I'm super excited about the research that she does here at USC, and we have been working on developing a model to simulate evolution of phytoplankton within ecosystem models. So when I fly here, this was my expectation, and I thought, nothing can really go wrong now, but this is reality, right? And even though I really wanted to be with you in person today and get to know you, we will need to wait for that. So thank you for coming here today. And since I arrived at LA, together with the lockdown, I will be presenting today my work that I did during my PhD. So I will talk a lot about mixotrophy today. And every time I talk about mixotrophy, I like to trace this parallel with binary systems. So we as humans, we have this tendency to classify everything as binary. But when we look closer, most of the things are not truly binary. So what about diversity, right? And on my case, on my work, I need to deal with the plant-animal dichotomy. So usually when we think about how living organisms will obtain carbon, we think about plants or animals. But what about organisms that can do both things? So many organisms that actually are mixotrophs. So if we have a closer look at the eukaryotic tree of life, you can see here that all the dashed lineages, they actually contain mixotrophic species. So mixotrophs are widespread. And this is particularly true if we look at mixotrophic protists. Most of them, they are mixotrophs. Most protists are mixotrophs. Here is just a schematic showing different plankton groups and how they occur from within a continuum from strict phototrophs to strict phagotrophs. And many of them are mixotrophs. So instead of thinking of mixotrophy as a curiosity, we actually could think that diatoms are the freak ones because they are the ones that seem to not have any mixotrophy within the group. And I just wanted to clarify that when I talk about mixotrophy, I am talking about the combination of both phototrophy and phagotrophy in a single cell. Now, why should we care about mixotrophic protists? So if we think about ocean biogeochemistry, it has been shown that mixotrophs, they can actually increase the production of VOC within a biological carbon fund. If we think about food webs and trophic ecology, mixotrophs could perhaps boost the transfer of carbon to upper trophic levels within the microbial food web. We also could think about mixotrophy as an intermediate step for us to better understand the evolution of eukaryotic life. And if we think about food security, many harmful algal bloom species, they are actually mixotrophic. So we need to better understand this strategy if we want to learn how to predict these events. And similar, we can think about environmental changes because mixotrophs might be a strategy that is advantageous once coastal systems are heavily eutrophic, or even the open oceans that are getting more oligotrophic and nutrient depleted with the warming of the oceans. But the main thing here is that protists are known to be important, but usually mixotrophy is overlooked. Now, mixotrophy is not a new thing. We know about mixotrophs for a while, and here you can see the first evidence of mixotrophic activity in the field. So this study on the left actually showed that nanoflagellates, previously thought to be strict autotrophic, they could actually consume bacteria. So we can think about these guys as plants that can eat, and they can have a significant impact on bacteria populations. But we also have, if you check here on the right, we have also ciliates that can contain functional chloroplasts. So on the other side of the spectrum, we can think about animals that can photosynthesize. So we know that they are occurring in the environment. And the dichotomy that we typically use to classify plankton or even describe them within ecosystem models, which is phytoplankton or zooplankton, might not be enough for us if we want to get a better understanding of their role within biogeochemical cycling in the oceans. We now know that there are many more complexities within the microbial food web, many ecological interactions, many black boxes that we are just starting to open now, and mixotrophy is one of them. So how can we think about mixotrophs? Because there is a problem when we think about them, because they are very diverse. They are not all equal. So in order to try to account for mixotrophic diversity, we can think about protists in a new functional way. So let's say we start with a protist here, then the first thing to ask is this protist capable of carbon fixation? If not, then we have those that are strict heterotrophs, like tympanids. But if yes, we need to verify if this protist is able to feed other plankton. And if not, we have the strict phototrophs like diatoms. But if yes, then we get mixotrophy. And now it's where we try to understand how diverse they are. And we could start trying to understand if they have a constitutive capacity for photosynthesis. Do they possess their own photosystems? If yes, we have here what we can call constitutive mixotrophs. And they can feed depending on the environmental conditions. Now, they are very diverse too, and I just wanted to show you some species that are constitutive mixotrophs. And here you can see like Carlodinium, all these small cells here are Carlodinium, and they can actually feed on very large zooplankton. So they have very different strategies too. Now, if we are with a protist that don't have this ability to photosynthesize, intrinsic ability, they need to acquire phototrophic machinery from their prey. And then we can call them the non-constitutive mixotrophs. So here are just some examples like Dinophysis, Noctiluca, Mesodinium, Strombidium. You can see here that they can be full of chloroplasts that they acquire from their prey. Then if we are dealing with acquired phototrophy, we need to investigate, can they acquire these classes from any kind of prey, or do they rely on a specific prey? So we could classify them as generalists if they can steal chloroplasts from different prey, or we have the specialists when they need a specific prey to acquire this phototrophic capacity. And those that are specialists, they can do that by stealing plastids, or they can also host an entire population of their phototrophic prey inside their cells through endosymbiosis. So by that, we can have 4 main mixotrophic functional types. And just to remind you that this will be important for you to get for the whole presentation now, their main ecophysiological differences are their innate or not capacity for phototrophy, the prey specificity required to acquire phototrophic capacity, and if they steal organelles or if they have endosymbionts. So by using this functional classification, I set on to investigate the biogeography of mixotrophs in the global oceans, and also the implications of mixotrophy to plankton succession and ecosystem functions. So this will be part 1 and part 2 of my talk, and I will finish just telling a little bit about what I am working at the moment in Naomi's lab. So for part 1 of the talk, I was interested first on investigating if mixotrophic protists, they were ubiquitous in the oceans, because we didn't have a very robust understanding about their how widespread they were. And also, if different mixotrophs have different biogeographies. So to do that, I first built a list of species that we know are mixotrophic. So from evidence from the literature, from experimental work, we can say, all right, these are the species that have mixotrophic potential. So of course, this will give us an underestimated picture of the biogeography of mixotrophy, because we simply don't know yet all the species that can be mixotrophic. And I also targeted studies which focus on mixotrophic populations. So by doing that, I compiled both qualitative and quantitative data. And first, I interrogated the OBIS data set, and I retrieved over 200,000 records for mixotrophs using OBIS. And then I also looked at published literature, and I got data for abundance, biomass, and bacteriovory of mixotrophs. And then I analyzed the data set according to 7 main oceanic biomes, and also following the classification of the ocean according to the Longhurst biogeographic provinces, as you can see here. So just to show you a map of their distribution around the globe. So here we have data for those that we can think as animals that can photosynthesize. So these mixotrophs, they need to acquire phototrophic capacity. And you can see here that they are everywhere. And here I compiled data for all 3 different functional types, but we can also have a look at the individual maps for each of them. And then when we do that, we start realizing that there are different distribution factors for each functional type, but also that they are biased, and we have more data available for the last group here, the endosymbiont specialists, and we also have a bias towards certain oceanic regions of the ocean. So what I did with this data set to analyze the data set, I built a matrix with presence and absence data for all the species across all the different biogeographic provinces. And then I did this NMDS. So here, each dot represents a different species. And then I grouped the species according to the functional classification for mixotrophs. And what is very interesting here is that we get 2 separate groups. So the generalist ones are very well separated from the endosymbiont ones. And the placid ones, they are somehow in a transition, with some species closer to generalists and others closer to this group. So one hypothesis for these differences in their distribution over across the oceans is how well they control their phototrophic machinery. And this control, we know that decreases from the generalists, it increases from the generalists towards the endosymbiont ones. So basically, these guys here, they usually can only stay with functional plastids up to 2 weeks without any prey. So they lose their capacity to photosynthesize very fast, and then they die. But endosymbiont ones, they can hold their functional plastids for much longer, even like over months, several months. So there is, these might play a role on their distribution in the oceans. So we also wanted to see what was going on in terms of biomass. We had less data available for biomass, but basically our objective here, first, we wanted to see how much of this bulk that we call microzooplankton, how much of it actually is represented by mixotrophic species. So we did that for 2 different groups, for ciliates and rhizaria, and we could obtain data for those species that are mixotrophic and those that are strict heterotrophic. So on this plot here, you can see the relative biomass of ciliates and rhizaria as mixotrophs. So 100%, we would have the whole assemblage of ciliates, for example, composed by mixotrophs. And as you go down, you have mainly strict heterotrophs. And first, the data is highly variable, so we cannot say that we have half in the oceans. It actually is very variable according to the time, temporal and spatial resolution of the data set. But what was interesting to us is that actually we saw a contrasting behavior for these groups when we correlated with the net primary productivity of the region. So basically this measurement is a proxy for nutrient load, and lower values, they represent more oligotrophic regions, and higher value, they represent more eutrophic regions. And what we saw here is that mixotrophic rhizaria, they tend to be more abundant within oligotrophic regions, while mixotrophic ciliates, they don't contribute as much. They tend to be very low, and then they increase in abundance as the environment gets more eutrophic. So depending on the environment, you might have a different mixotrophic community. And again, this can be related with how well they control this acquired phototrophic machinery. So now I will be talking about those that are plants that can eat. So here is a map for the constitutive mixotrophs, and again, they are everywhere in the oceans. We can also have a look for the data across different taxonomic groups, and then we have a lot more data for dinophytes here, and it seems that we don't have a lot of mixotrophs on the other taxonomic groups. This is what the data is telling us, but you will see that's not the reality. So if we check for different size classes, we have a lot more data for cells that are larger than 20 micrometers, which are mainly dinoflagellates. So this gives us this idea that mixotrophs are mainly dinoflagellates in larger cells. But is this really real? So we plot here all the data from OBIS for all mixotrophs smaller than 20 micrometers. And this is the distribution pattern that we got. And then we compare that with data from studies that targeted mixotrophic populations on the same size fraction. So we have less data here. There are not many studies out there that did it. I think it's around 22 papers that data come from. And you can see here a big difference when we think about oligotrophic regions. So just looking at OBIS, you wouldn't really capture the presence of these mixotrophs within these regions. But when we look at these studies, we start filling the gaps. So this is very important, not only because these mixotrophs, they are present in these regions, but because they are active. So they are actually consuming bacterial populations. So here are some data that I compile as well across all the studies. And you can see data for the different oceanic biomes. And what you see here is the ratio between the bacterivore impact by mixotrophs to the heterotrophic nanoflagellates. So basically, if we have all the dots falling here at 1, would mean that both mixotrophs and heterotrophs, they had the same impact consuming bacteria. So you can see here that mixotrophs, they are active consumers of bacteria and across all these different oceanic biomes. So they are active and they have an impact within the microbial food web. And just to compare that, recently there is another study that did the same biogeographic analysis according to the functional type of mixotrophs, but using data from tidal oceans, metabar coding data. And they also found that these mixotrophs, they are very important across the oceans and particularly within oligotrophic seas. Sorry. So that takes us to the end of part 1. And the key message here is that mixotrophy is ubiquitous in the global oceans, and that different mixotrophic functional types, they can have also different biogeographic distributions. And global data are strongly biased by science, taxonomy, and oceanic region. So the goal now, the challenge for the community is to use methods that can actually target mixotrophs. It's very complicated, and usually these studies that go there and actually target mixotrophy, they usually lack a more taxonomic resolution of the community. So we know that mixotrophs are active, but we don't really know who they are. So the challenge now is to combine different methodologies to be able to fill these gaps and gain a better understanding of mixotrophic nutrition. So this takes us to part 2 of the talk. And now I will be talking about the implications of mixotrophy to plankton succession and ecosystem functions. For this part of the talk, I will present my work that I did using ecosystem models to gain an understanding of the role of mixotrophy within the microbial food web. So the idea here is that we can simulate a biological community by assigning different plankton functional types. And then this community is subjected to environmental forcing, and then we will see changes in the community, and these will reflect changes on ecosystem functioning. So the model that I use is a flexible model to represent different protists. So using this model, you can switch on or switch off certain properties, and you can simulate strict phototrophs, strict heterotrophs, or mixotrophic protists. So this model is a stoichiometric model, and then you can account for carbon, nitrogen, phosphorus silicate, and chlorophyll content within the cell. And all these quotas, they are variable, so it's an emergent property of the model. And here I'm just showing a schematic, a schematic for a phytoplankton cell, so strict phototrophic protist. And then within the circles, you can see the different processes that are being simulated. And the squares here, they show the nutrient pools, organic and inorganic pools within the model, and how they interact. Oh, sorry, I think they're doing some construction here right now. I hope this won't be a problem. All right, so I won't show you all the equations, but I wanted to show some key equations that drive the dynamics of the model. So here we have the nutrient status of the cell. So this is very important because it's based on the stoichiometry of the cell, and they can have a minimum and a maximum value, and the nutrient status of the cell will vary. So as closer to the maximum value, it means that the cell is with a high nutrient status. And as this value decreases, then the cell is stressed. And we also have a term computing light limitation, which will depend on the initial slope of the photosynthesis radiance curve, the chlorophyll to C quota of the cell, and also on irradiance levels. So on the model, photosynthesis depends on the nutrient status of the cell and on light limitation. Nutrient uptake will depend on external nutrients through a Michaelis-Menten type equation, and also on the quota saturation. So as the quota gets full, nutrient uptake is blocked. And respiration will have two components. One component associated to basal maintenance costs, and another one associated to biosynthesis, which is linked to nutrient uptake. So now this is a schematic for a strict heterotrophic protist. So now we are simulating different processes here. And what is important here to this model is that we have a satiation feedback that will impact the ingestion of prey. So basically, we assume that the protist has a maximum capacity within the feeding vacuums, and as this feeding vacuole gets full, they will be able to control the ingestion rate and block it. So the ingestion will depend on predator and prey encounter rates, will depend on the satiation feedback, and also on a size selectivity term. So basically, ingestion rates are maximized if the prey is of the optimal size, and then this will decrease as the prey deviates from this optimal value. Then we also compute prey digestion, and these will depend on prey quality and quantity. So assimilation efficiency is an emergent property of the model too, depending on these two features. And respiration, again, has two components, one related to basal costs and the other one associated to the assimilation of carbon from the prey. So how to simulate mixotrophs then? If we are thinking about those that are plants that can eat, we can start with a setup just as for the autotrophic protists, but now we can allow the ingestion of prey. So how can we do that? So basically, phototrophy will now affect digestion rates. What we say is that if enough carbon is flowing from photosynthesis, there is no need to eat that much. But as photosynthesis decreases, then the need to ingestion increases too. So basically we have a term here, PRAL, that is the ratio between the actual photosynthetic rate and the maximum photosynthetic capacity. So this will kind of regulate how mixotrophic activity is simulated. So this is a critical, these are the critical equations describing mixotrophy. And also we have a penalty because we assume these mixotrophs, they cannot grow strictly heterotrophically. So they need a critical proportion of growth flowing through photosynthesis, otherwise they die. And another important assumption of the model when simulating mixotrophy is that mixotrophs, they are more efficient utilizing nutrients. So as they can get nutrients from their prey, they can also combine these nutrients with carbon that was just fixed through photosynthesis, and then they can re-assimilate ammonia internally. So basically what this is saying is that instead of just remineralizing nutrients back to the environment as a strict phagotrophic protists would do, they can actually reutilize it and avoid releasing these nutrients back to the environment by combining phototrophy and phagotrophy. And now the costs of being a mixotroph, they go down to respiration. So they have two additional costs compared to phototrophic protists. So now they also have a cost associated to the assimilation of carbon from their prey, and also by doing this internal re-assimilation of ammonia. And if we think about those that are animals that can photosynthesize, these guys, they need to acquire their phototrophic capacity from their phototrophic prey. So we assume that they do not photoacclimate, and we also assume that their plastids are diluted at a constant rate over time. So these are the main features of the model. And then using this model, we wanted to understand how mixotrophy would impact nutrient cycling and trophodynamics, and also how different mixotrophs they would respond to light or nutrient limitation. So to do that, we simulated a microbial food web, and in order to test our predictions, we first built a food web that has no mixotrophs. So here we use the plant-animal dichotomy. So we just have autotrophs in green here, and heterotrophs, and also decomposers. And each schematic here represents a different functional type in our model. And as you can see, size increases from the bottom to the top. So we have this setup for the food web, and then we assume certain ecological interactions. So nanoplankton, they will be able to feed on phytoplankton and on the same size clusters there, and on nanoplankton. Microplankton will be able to eat on phyto, nano, and micro. And copepods, they can prey all functional types except for phyto prey. So these are our assumptions, and we also consider intrabute predation. Now, when we wanted to represent mixotrophs, I will show here. So what we did, we considered nanoflagellates and microflagellates, mainly dinoflagellates, that were previously thought to be phototrophic. We now consider that these two groups, they are like plants that can eat. So they are mixotrophs. And they have a set of ecological interactions, which basically we assume that they can prey on the same prey of their heterotrophic competitor with the same size. And then we also consider the addition of two extra functional types within microzooplankton. So these three groups, they kind of represent microzooplankton, and now we have animals that can photosynthesize, and we wanted to describe two types. So one are the specialists that need to acquire phototrophic capacity from specific prey, and the other one are the generalists that can rely on diverse prey types. So the main difference between these two groups is that we assume the specialists can also take up inorganic nutrients, and they have a lower dilution rate of their plastids compared to the generalists. And they are able to prey on the same prey types as their heterotrophic competitor. So using this food web, we did some chemostat modeling experiments to start with. So we have here a homogeneous box with 10 meters depth, and this box is subjected to a dilution rate, which acts bringing new nutrients to the system and also washing out nutrients and plankton biomass. And then we did here, we run two different limitation scenarios. So the first one, we assume that light was limiting phototrophic growth. And then on the second one, we assume that nutrients were the limiting factor. And then we wanted to see what happened with our food webs. So here I'm showing you some results for the cycling of nutrients. So here in the y-axis, you have the regeneration of ammonia in the system, both on the non-mixotrophic and the mixotrophic food webs. And as you can see on both limitation scenarios, we have a decrease in the regeneration of ammonia once you assign mixotrophic protists. And if we look at the trophic transfer efficiency, we actually have an increase in the trophic transfer on both scenarios where mixotrophs are considered. So why is this happening? I'm just showing you some results for the nutrient limited scenario here because this was more pronounced on this scenario. So you can see here the schematic for the non-mixotrophic food web. And on this scenario, what happened is that we got the exclusion of 3 phototrophic groups. And the main groups supporting primary production were the phytoplankton fraction, which are not as accessible to copepods as prey. So copepods need to rely mainly on heterotrophic prey to grow. And also on this system, we have here these 2 groups responsible for most of the remineralization of nutrients in the system. Now, once we have mixotrophs, we saw something very different. Actually, we got the exclusion of these 2 groups now. All mixotrophs could exist in the model, and the biomass within larger size classes increased a lot on this scenario. So mixotrophs, they actually allow the accumulation of biomass in larger size classes, and they avoided the explosion of phytoplankton biomass in the model, and this supported higher trophic transfer efficiency. And now that you have a lot of mixotrophs in the model, you don't have as much of ammonia being regenerated in the system because they can do this internal recycling of nutrients. So this capacity can have an impact and even increase nutrient depletion in the system, which can make it more difficult for strict phototrophs to thrive. We also wanted to check the succession of different trophic strategies across a gradient of light and nutrient limitations. So here we have nutrient availability increasing in the y-axis, and here we have light increasing in the x-axis. And what was very interesting is that mixotrophic nanoflagellates, they actually dominated during nutrient depleted conditions and highlight, which is expected for this group, while mixotrophic nanoflagellates, they had their highest contribution when light was limiting but nutrients were available. So we can think about it when we go to the sea and we actually measure mixotrophic activity. These guys seem to be very important in oligotrophic systems, while these ones could actually thrive in coastal eutrophic environments, which usually are limited by light due to the high turbidity in these systems. And we also could see an increase in autotrophs along this gradient, which is what we would expect. Now, when we see the heterotrophic competitors, so we did the same thing here, but now for heterotrophic protists and those mixotrophs that are like animals that can photosynthesize. So we actually saw that generalists would dominate more once nutrients are depleted, as expected. But for the specialist ones, it was not very clear. We know that, for example, Mesodinion is a species that can thrive during nutrient replete conditions, but light limited. But that's not what we saw in the model, which indicates that we are missing some important trade-off that can represent the niche differences between these mixotrophs. But again, we saw an increase on the heterotrophic population along this gradient, which is what is expected. So this was all a theoretical modeling analysis, and we wanted now to see how our model could explain observational data. And by using long-term time series, we wanted to answer if differences in necrophysiology could explain the succession of different mixotrophic types. And also if the identity of the mixotrophic type would matter to set carbon fluxes in the model. So we did that by using data from L4 station, which is located in the Western English Channel. So it's a coastal temperate sea. And I used a time series of 9 years, and data were collected weekly. And in order to represent environmental variability at this site, I coupled my model to Gotham, which is a model that resolves the vertical structure of the water column. And I was able to represent 100 vertical layers to simulate the dynamics, the plankton dynamics at L4. So I was interested on the seasonal progression of plankton dynamics. So here you can see some results of how well our simulations, they match observations. And what you can see here is the climatological averages over the months. And on the x-axis, you see different properties. On this case, we have nutrient data. So the dots, they show observations, and the lines, they show the simulations. And the results from my model, they are the blue line, and in yellow, you can see results for another model, which is AirZen, which basically has the same food web setup that my mixotrophic model, but they don't describe mixotrophy. And we just wanted to compare our modeling results with AirZen to have an idea of how well we can simulate data compared to a model that has been used across different oceanic regions. So as you can see, our model does very well representing nutrients overall, and also total chlorophyll and total plankton biomass. It actually performs as good as AirZen, or sometimes even better. So AirZen here overestimates total chlorophyll more than our model does. So we were pretty confident with these results. But what I was interested was on evaluating the different trophic strategies of protists. So here we have data for autotrophs, mixotrophs, and heterotrophs. And this is all from my model, my mixotrophic model. So overall, it was very good for these three strategies. And then I wanted to look at the different mixotrophic functional types. So in this model, we have here mixotrophic nanoflagellates, mixotrophic mainly dinoflagellates, and mixotrophic ciliates. So I was very happy with these results and this one, and a little bit frustrated that the peak of dinoflagellates was off. So we didn't get this timing right. And the main issue here is that the model has a limitation to describe the dynamics from winter to spring. So basically what happens at L4, it's very particular of this region, is that mesozooplankton, they can appear first, then their prey. And this happens probably because mesozooplankton is being infected from somewhere else. and we are not representing advection in our model. So we don't simulate this mesozooplankton peak that early, and what happens is that we have an early peak of autotrophic biomass. They respond earlier than expected, and they grow more. It's a little bit overestimated compared to the data. And this is because there is no mesozooplankton to control this bloom. And why is this related to this peak here is because in the model we assume that mixotrophic dinoflagellates, they can feed on diatoms. So that's why it's earlier than expected, the bloom. But still, we were interested on evaluating the seasonal succession of mixotrophs, and I will show you that the mixotrophic dinoflagellates were the key to understand the dynamics of this system. So now I wanted to use my model to try to understand the seasonal succession of these groups. And usually, mixotrophy is thought as a strategy to be used during summer in temperate seas. So here is just an example that I like a lot. Is a comic from Robert Fisher. So you have here an Ochromonas in the water column, and it's enjoying the nutrients in the sun. And then you have a bacterial population here. And over time, these nutrients, they get depleted in the water column. And at some point, these Ochromonas will just capture all bacteria and feed on them. So this is what we typically view as a mixotrophic strategy, to be good when nutrients are depleted. But with this model, what we saw is actually that mixotrophy is important at all times in the seasonal succession and also over depth. So we saw their importance during summer, but also at other times. So here, what you can see is data over depth and over the seasonal cycle. And I have data here for total protist biomass. And I wanted to evaluate the relative contribution of these different trophic strategies at different periods of the seasonal cycle. So as you can see here during spring, we have a higher contribution of autotrophs as expected, and during summer, their contribution decreases and you give space for mixotrophs, which again was expected. But something that we also saw with this model is that mixotrophs, they can contribute a lot at late winter. So mixotroph is also a strategy that is important once light is limiting. And this was less explored so far when compared to nutrient limitation on mixotrophy ecology. So this was very interesting. And this happens because mixotrophic activity in the model is not a fixed property, it's actually an emergent property of the model. And you can see here a proxy for mixotrophic activity. So basically, again, we have the seasonal cycle here, and on the y-axis, we have the relative phototrophic growth. So it's basically the contribution of phototrophy to the total growth of different mixotrophs. So as this value increases and gets closer to 1, this mixotroph is growing purely phototrophically. And if this value decreases, we have here a much higher contribution of phagotrophy to growth. So as you can see, we have different trajectories for different mixotrophs. And actually, when we look at mixotrophic nanoflagellates, they are utilizing phagotrophy quite a lot during winter too. So this is interesting that with the model, we can actually simulate mixotrophic activity and see how this varies over the seasonal cycle and for the different types. So with that, we went out to try to understand the role of different mixotrophs setting carbon fluxes in the model. So to do that, we averaged the carbon flux over the seasonal cycle. So that's what I'm presenting now, averages. And we investigated first the gross primary productivity. So here you can see the results for my model with all the mixotrophic types. And we have data, GPP data for different size fractions. So phytoplankton, nanoplankton, and microplankton. Now, if we run the same model, but with no mixotrophs, that's what we see. So now you can see that biomass is accumulating here in the picofraction, and we don't have as much biomass in larger size fractions. And we got a very similar result for all the runs that did not account for mixotrophic nanoflagellates. So in all these runs, phytoplankton biomass was very high, and therefore they contributed a lot to GPP. Now, once you have only mixotrophic nanoflagellates in the model, you basically have them outcompete the phytoplankton fraction because they can also feed on them. And then you accumulate biomass in larger size fractions. And these are just other results for different combinations of mixotrophic types in the model. So the main message from here is that it really depends on the mixotrophic type that we are looking at to understand how mixotrophy might impact the carbon flux. And here, mixotrophic nanoflagellates were key to support this higher contribution of larger size classes to total GPP. And this has been suggested already by a previous study by Ward and Follows. So they also, their modeling study, they compare two food webs, one which is in blue here that has no mixotrophs, and the other one in red, which they assumed all plankton to be mixotrophs. And they also saw that biomass would accumulate in larger size classes. What we are doing and extending on that is that it really depends on the type of mixotrophy that we are looking at. So the identity of the mixotrophic community is important. So we also wanted to see other carbon fluxes in the model. So here you can see a schematic. Our control model has all these three different types, and then we evaluated bacterivory, herbivory, VOC production, trophic transfer, and vertical export. And just as an example, I will guide you through the results that we got for the trophic transfer. So what I'm showing to you here is the percentage of change once we have no mixotrophs in the model. So trophic transfer decreases substantially once we don't have mixotrophs, as expected. And this was also observed when you only have this type of mixotroph, or only this one, or a combination of these two. So then these results, they hold. But once you have only mixotrophic nanoflagellates, you basically don't see any change. So they are the ones driving the dynamics of the model. And they are the ones facilitating the accumulation of carbon in larger size classes, and therefore favoring trophic transfer efficiency. Now, what I wanted to show you is that here on this version, we actually see that there is a little increase in the percentage of change. So once we have in the model only these two types, we actually see that trophic transfer could increase because it really depends on the trophic interaction. So mixotrophic dinoflagellates, they have a tight relationship with diatoms in the model. They can consume diatoms, and therefore diatoms are not available for copepods as much as they would in the absence of this predator. So that's why we see a relative increase here, because we have now more diatoms that can boost trophic transfer. So the main message is that the impact of mixotrophy on carbon fluxes would depend on the identity of this community. So I'm not going to details now, but we also saw that for all the different carbon fluxes evaluated in the model, that depending on your community, you can have a different impact. So in the case of L4, for example, mixotrophic ciliates, they were not so abundant, and therefore mixotrophic nanoflagellates were driving the dynamics of the model. So with this, I finished part 2, and I just wanted to say that the main outcomes from this work was that differences in physiology can explain the succession of mixotrophic functional types. The identity of the mixotrophic community is important to understand their role in carbon fluxes, and we still have many open questions. So it's very difficult to explain the coexistence between different mixotrophic types and even their strict autotrophic or heterotrophic competitors. This is very tricky to keep in the model. So it shows some avenues for future research that can help us to better parameterize the models and to understand their communities. And we still don't really know the role of mixotrophy on community stability, because because of this diversity. They are just too different. We need to start assigning this diversity when we talk about mixotrophs. So this takes me to part 3. I'm not sure how I am with time, but I will be very brief here. So I just wanted to share with you what I am working at the moment. So I came to USC with this challenge to simulate phytoplankton adaptation within ecosystem models. And luckily, Nate and Naomi, they have been working on it for a while, and they have been developing new modeling frameworks so that we can do that. And the novelty about their framework is that they are not only focused on the evolution of independent trait values, but they are looking at trait correlations, because certain traits, they will not evolve independently. And it is important to account for their relationship. So when we think about ecosystem models and how plankton dynamics are described within these models, this is very important, because what we have a certain set of key trade-offs that represent competitive advantages between different phytoplankton groups, for example. And these trade-offs, they are fixed in the models. But what if we think that these trade-offs, they can actually be broken through evolution? So what would happen? So my work now is to combine this type of model I just showed to you today with the modeling framework that Nate has been developing, so that we can allow for the evolution of these trade-offs. And what we are looking is to find the key trade-offs to focus, and also how we could use these models to try to understand the possible evolutionary trajectories that phytoplankton populations might take to adapt to environmental change. So there are many cool unknowns to be explored. I am very interested now on the temperature dependence of phytoplankton metabolism. So I'm looking about how photosynthesis and respiration are sensitive to changes in temperature, and how different they respond to temperature. And at some point, I hope that we can bring also mixotrophy to our studies, since phagotrophy will also be affected by temperature in a different way. So that's what I'm doing at the moment. And having said that, I finished my talk. So I wanted to thank all my supervisors during my PhD and the people that collaborated with me for the biogeographic work. Also thank my funding agency from the Brazilian government, and also Kino for Mafalda, that always makes my presentations a lot more fun for me to make them. Unfortunately, I just learned that he died. He passed away last week. So if you don't know Mafalda, just run and go check it out. She's awesome. And a big thanks to the Levine Lab. I received a very warm welcome when I got here in March. Unfortunately, that's how we have been interacting now. But even though the smiles and all the support are helping a lot to go through this pandemic. So thank you all for listening. And now I will take any questions. Thank you.
Thanks, Suzana. I think if anyone has questions, you can either pipe up directly or type it in the chat. Either, I think, are you able to access the chat?
I'm checking this now.
Okay, otherwise I can read through it.
Yeah, I don't think, maybe if I stop sharing, I can, but.
Yeah. I had a quick question for you. I was just wondering, it seems like a lot of these mixotrophs, and Noctiluca might be one example, but some of the flagellates as well, they're also characterized by toxicity. So they don't necessarily, I'm just wondering if, in fact, it seems to me that that's rather widespread amongst a lot of these taxa. And I wonder if that would tend to work against the idea of mixotrophy enhancing trophic transfer. Noctiluca is the one that comes to mind. I think you generally don't associate it with being a windfall for copepods when there's a Noctiluca bloom. So I was wondering if you could elaborate on that.
No, you are totally right. Actually, that's why it's difficult to generalize and say how mixotrophy will impact the trophic food web. Toxicity is something that it's completely absent from our models in general. So it's something that will be very interesting to look at and to develop. So as you mentioned, like Noctiluca, there is a work in the Arabian Sea where it shows that Noctiluca, a mixotrophic one, would actually, with the changes in climate and everything, and also with the coastal trophication of that area, they were replacing diatoms bloom. They would feed on them and also replace them. And they were actually causing a massive fish killing, I guess, in the area. So they were having a negative impact in the food web. So you are totally right. And I know about some work from Claudia Traboni and Albert Calbe in Barcelona, that they were experimentally testing if mixotrophs, they could actually enhance the gross growth efficiency of copepods. So they tested different prey types, so autotrophs, heterotrophs, or mixotrophs, and they didn't find evidence for that, for an increase on GGN. And they related that mainly to the toxin production. They were testing carlodinium. So carlodinium is toxic, and then they saw the opposite, that these mixotrophs, for being toxic, they could have an opposite effect. So you are totally right, and we just don't represent that in ecosystem models, so this could have an impact too. Again, it goes down on which type of mixotroph we are talking about. I hope I have answered.
Yeah, Trunkai, did you have a question?
Oh yeah, sure. So so Suzana, great talk. I really enjoy listening. A question is that, so basically all the mixotrophs and the model that you build up for the protist, they are all simplified coarse-grained model in terms of their pathway. And then obviously, I mean, in our department, there are many different labs. They are studying about the cell biology of different kinds of bacteria or different kinds of protists. What are the key information that you want to get from those people who are carrying out physiology study?
Yeah, from lab experiments, from culture, or from the field, or from both?
Well, in general, from both.
Yeah. So I think it's very promising to validate these models, this information, like it's done here at USC, right, in the Dave Caron's lab for gene expression. If we can target, I think molecular tools, they have a great potential to help to understand mixotrophic ecology. So if you target genes that are related to phototrophic nutrition or phagotrophic nutrition, and you can understand how these vary over different environmental gradients, I think this would help a lot to understand that. So all the different metabolic pathways, even because sometimes it's not clear which mixotrophs they can actually take up nutrients or how much they do and how much this contributes to their growth. So this would be very key to better parameterize these models. Know this proportion, how this proportion of phototrophic phagotrophy varies and contributes to growth. And from field, this can be done with cultured species. It's more complicated if we think about field samples. I don't think you can readily do it. I'm not an expert on molecular tools, not at all. Probably there are many people here that can say more about it than me. But in the field, I think it's more tricky, especially if we want sequence data. There are far more OTUs than species, right, described. And it's difficult to compare different studies. So we don't get the information of who is mixotrophy. This information is not readily available. So one way for field samples is to use methodologies that can actually verify mixotrophic activity. So some methods, they can actually stain the feeding vacuums. It's very simple concept. So you could collect a sample and then use this marker that can stain the feeding vacuum. Then you can at least know the proportion of the community at that point in space and time that was actively feeding. So this is a measurement of that, but we need to combine that with high resolution taxonomy too, which is not usually done. So I think this would be a great, would provide very great information for us, because the analysis that I showed to you at the beginning, they are interesting, but they, of course, they compile data from different studies, different methodologies. So it's difficult to analyze. So if we have a protocol that is the same protocol is applied to different regions over long time periods, and then we can have a better idea of mixotrophic ecology.
Yeah, thank you.
Thank you.
Great. So I think we're at 1 o'clock. So I want to make sure you get a break before you have to meet with a group of grad students. So for everyone, you're good to go. Thanks so much for your time and for giving the talk. And for everyone who's hanging out with the grad students, for the grad students who are hanging out, we're going to use the same link. So let's just take 15 minutes and come back at 1:15.
All right. Thank you, Nina.
Thank you.